We'll move with that to our fourth working of our initial working groups. And to present that, we've got Kathryn North from Australia. And she's flying solo because Charles Sawyers can't be here.
Thanks, Peter. Could I have the slides for the clinical working group? Thank you. So the clinical working group has a mandate and a goal, there we go, to look at how we represent phenotypic data and link it to genotypic information. I know that the data group considers that we deal with metadata, but we are managing to define all the different types of metadata as we go along. We decided to start with a relatively low-hanging fruit in the field, which is rare Mendelian disorders and also cancer because of the immediate therapeutic implications. And I think it's important to go through the approach that we've taken. Primarily, we wanted to make sure that we avoided reinventing the wheel and that we're adding value to the many existing initiatives and endeavours that are out there. We're looking at addressing both research and clinical use scenarios, and we're doing what we're doing really with the physician in mind, but taking into account that it's going to be a continual iterative process between what we're doing in research and what we're translating into the clinic, at all times with the centre being the patient. So this is a list of our clinical working group members, and it expands exponentially with the addition of our various task teams. And as Peter's mentioned, I'm representing myself and Charles Sawyer today. And again, we have complementary expertise. My areas are in neurology and clinical genetics, and Charles within oncology. And I must emphasise that for all of our different working groups, our coordinators are essential to everything we do, and Farah has been fabulous in supporting our team and getting us all on the telephone calls at dreadful hours of the day and night. So our approach to the various problems is listed here, and this was actually initially the brainchild of Heidi Raim, is that we need to define the need in the area, document the existing efforts, look at the compatibility of approaches that exist, identify the gaps, see how we can fill them, and then try and do that with a demonstration project, looking at implementing probably initially in a research and development environment before we then translate and integrate in clinical use. Our long-term goals, of course, are to be continually adaptable and, as has been mentioned in terms of all of our endeavours, to communicate globally so that we're starting to get the message out there. And what I thought I'd do is just go through where we're up to in all of these different areas. So this is now the web has been, our website's been rapidly populated over the last couple of weeks leading up to this meeting. And these are the task teams that are associated with the clinical working group. And I must say, I think obviously clinical working group is incredibly broad, and I see our role as being to, initially at our working group meeting in March, to identify what we saw really important areas. And then we've acted as, I think, to facilitate to get things going across a number of these different areas, and then act as an umbrella in terms of driving things forward, and as well as identifying new initiatives in which we wish to be involved. And you're going to hear specifically about the Breast Cancer Challenge and the Matchmaker Exchange Program. And what I thought I'd do is just quickly go through, in a slide each, what we're doing under each of these initiative headings. So first of all, the catalogue of activities, which I called the Mapping Initiative, but the name got changed to go onto the website. And the aim of this was really as a starting point, as working out what's going on out there. And that ended up being quite a huge task. We've started with Mendelian rare disorders and really then pulling together a list of all the different initiatives that are happening as a resource for researchers and for clinicians, and to also identify particularly what we might integrate, for example, into the Matchmaker Exchange project. We've completed the Mendelian genetic disorders. It's been posted on the website, Genomics and Health. And the next topics that we're tackling in terms of mapping are in the areas of what's going on in electronic medical records in e-health, as well as what's happening in cancer. So this is from the website. It's probably difficult to read, but just to give you a feeling for this, is it's a living document. So when you go onto the website, and if your particular consortium or the tool that you've developed that's available on the web or your database is not available, then just contact us, and this is a continual living document that we will update. We currently have data on over 40 different initiatives. These are the large initiatives in the areas of rare disease, and you can search depending on your interest. You know, here we've clicked on Mendelian disorder and consortium, and then you'll get a list. There's 15 different rare disease consortiums that we have data on. We put up links to their website and contact people. So it's really so that you can see what's out there related to your field, and also if there's disease-specific areas of expertise. So we want this to be a useful focus. I must say for myself, it was an incredible learning experience just seeing the breadth and the wealth and the depth of work that's going on in the area. And that's, I really think, what every one of the working groups is aimed to do, is try to sort of link into that and bring everything together. So the BRCA challenge, we're going to hear from John and Stephen in the next session. And the goal of this is to aggregate BRCA1 and BRCA2 data to really support research activities and to generate the most informed understanding that we can of BRCA variation and its impact on human health. And the goal of this group is really to look at consolidating again what is out there. There's a huge amount of work and great expertise internationally, and in the longer term to expand those data sources. BRCA was first discussed at our March meeting in London and then expanded upon at the Human Variome Project meeting in Paris. And what John and Stephen have done, and the steering committee for that group, is to bring together many of the international experts that are working in this field. This is quite an exciting project because it's something that will certainly capture the public imagination as well as providing incredibly useful clinical information. And we really see it as creating a model of how we might approach large data sets on specific disorders. The Matchmaker Challenge is led by Heidi, Anthony and Kim. And the challenge here is, as we know, that we've got data collected in many silos. We've also got the problem that currently, if you're doing clinical exomes, greater than 60% of exome sequencing cases are unsolved. The case data tends to sit in isolated databases and it's not readily accessible. And phenotypic data is underrepresented in most of these genome databases, so it's very, very difficult to link. The problem we imagined was if you're a clinician or a researcher sitting in Melbourne, Australia, for example, and you've done an exome, you've found what you think is a highly likely disease gene, but it hasn't been described before, and you find a variant. How can you go in and then query a number of different databases to see whether other people have found variants in the same gene and then in patients with the same phenotype? This is really replacing what I like to say emailing your mates, which is what you usually do when you find a potential disease gene. On the other hand, there's also the aspect of if you've got a particular complex or rare phenotype and you're looking for other patients with the same phenotype, how do you bring them together? And so Matchmaker Exchange, which Heidi is going to go into, is addressing that particular issue. Now, genomic data and electronic health records, this was something that we brought up in March as a long-term goal. I think we've already heard about today how the electronic health record is one of the banes of all of our existence, and how we can link up information and feed it across different institutions. The challenge is, how do we realise the value of electronic health records? In some places it's how do we actually get electronic health records and use them to contribute to genomic medicine and as research tools. And there have been a number of great studies already that show the power of the electronic health record. And so recently, led by John Maddison and Andrew Morris, we've formed quite an active and actually a positive task team that is starting to look at how we address those challenges. And again, the first need was to map the existing and emerging initiatives. And this is very much done in partnership with the G2MC, as it's called, or the NIH-formed Global Genomic Medicine Collaboration. And then these are a list of the various organisations that are very active in the field that are now getting together on regular phone calls to start to address specific bite-sized chunks about how we can make this work. It's also been great because we've identified, as part of the Global Alliance, various institutions internationally that are at the same phase of rolling out electronic health records, where we can start to do test cases of integrating genomic data into the records themselves and compare that across sites, which I think is going to be extremely strong. The clinical cancer genome really it started off starting to look at actionable variants that are guiding therapies in cancer. And this is led by Charles Sawyer and Mark Lawler. And the goal of this, and really the first, this has been the subject of a newly formed task team and then bringing together a group of people tomorrow. But the goal is, again, to harmonise the many, many clinical sequencing efforts in the global cancer community. Again, to map those efforts, to share data, and promoting the standards and best practice supporting decision making. And tomorrow at our clinical working group, we're going to hear about different perspectives from Singapore, Europe and North America. The immediate goals are to consolidate a table of actionable genes and also the development of panels and the recommendations in those areas. Again, we'll be talking about and taking note and working with existing initiatives in the area, which I've listed here, and we certainly see the opportunities to harmonise, and particularly I think between them Charles and Mark are involved in most of those organisations. I'm going to end with phenotype ontologies, and it was particularly something that we talked about a lot yesterday at the data working group meeting. And really, there's been a huge amount of work in this particular area, supported by a number of the international organisations that are listed in our catalogue of activities, but particularly the IRD-IRC and HVP. And this effort is now led by Peter Robinson, who I think is here. Hi, Peter. And based from Germany, who's really been, the leading figure in terms of human phenotype ontologies. And the aim of what we need to do here, and I think it's going to be a big focus for our clinical working group in partnership with the other working groups, is really bringing together the international efforts to develop and promote a standard language of how we record patient clinical phenotypes. And it's not going to just be the clinical features. We realise we need to incorporate biomarkers, imaging, a lot of things that aren't already included in some of the classic ontologies. And this is actually a slide from Peter, is that, a football's not a football is not a football. It's you need to really be describing specifics, particularly in something as complex as human clinical disease. So I'm going to give you an example of the approach that we're taking with the phenotype ontologies, and just to run through this slide, which you can't read. So it's really going through what our initial approach was. We defined the need. So we need to have a standard and structuredized phenotypic data with which we can link all of the genomic and other metadata. We initially, as I've mentioned, documented existing efforts. The human phenotype ontology is like the concise Oxford Dictionary of ontologies. It's very comprehensive and many, many thousands of terms. And then what has developed is there are other also interusable and compatible different ontologies, and also databases that are using that are searchable diagnostic tools. And I've listed, this isn't an exhaustive list, but it gives you an idea of the range of different classifications in the areas, including SNOMED, that's used in electronic health records, and the ICD, which is commonly used in terms of coding patients in hospitals. We've also got to think about linking in with things like OMIM and OrphanNet and PubMed, and there are a number of tools that we found that are also supporting data collection using these ontologies. So it's unlikely that a single ontology is going to support all needs, and certainly Peter and Ada Hamosh have led a group internationally to look at the most commonly used terms in human phenotype ontologies and to map them and to look at supporting interoperability, and have come up with a core definition of around 2,500 such terms that are used. And currently, Peter is now there. We found that there were, I think, a couple of different. I think it was 3 or 4 different working groups that were looking at phenotype ontologies, and now they're all brought together under the leadership of Peter Robinson. So finally, the important thing now is rare diseases are pretty well covered, but there were, even at our March meeting, a number of gaps that were identified, such as neurodegenerative disorders, for example. But here I use the example of cancer because I think it's one that we need to develop quickly. There are a number of existing terminologies, but the challenge is really that we need to integrate these existing ontologies. We also need to look at how we capture a range of data: pathology, cell surface biomarkers, imaging, and longitudinal data on an individual patient, because it's going to change over time. And then looking at how we, include these phenotypic aspects and link to the genomic aspects of particular tumours. So that's really a big area for work that we'll be looking at doing within the clinical working group and in combination with the data working group. So I'm going to end there. We have our working group meeting tomorrow, which is fully subscribed. But really, we're focusing on a number of these areas that I've been talking about, and particularly looking at our cross-cutting projects, because part of what we also need to do is bring our different working groups together with a lot of cross-talk, because all of the issues that we're dealing with have implications across the working groups. So thank you.
So we'll follow exactly the same format as before. So if anyone's got a question for Kathy, if you can come to the microphone, identify yourself. I'll try and see you against the lights from the back. And we've got probably about 10 minutes for questions. I'm trying to keep us as far behind as we were when David handed over to me. And then we'll move, after this session, we'll move into those projects. So I would say if your question relates to those projects, it may be good to save them till we've had the project presentation. But if it was anything else that Kathy discussed, electronic health records or the rest of the talk, now will be the time to ask those questions. While I'm waiting for the first person to come to the microphone, Kathy, we mentioned right at the beginning that there was a focus on cancer and rare diseases, but I wondered if you could just talk about where we start and where we think we need to go eventually.
Absolutely. So we really, as I said, we tried to look at the low-hanging fruit initially and where there's a lot of activity. But obviously everything that we're building is looking to then move into complex disorders and a much more population health base as well. But it was really aiming to start in quite focused areas. And also I think the area of cancer is probably the one where there is most immediate relevance in terms of targeting therapeutics. And in rare Mendelian disorders, which many of these are cause of chronic intellectual or physical disability, the ability to provide prenatal diagnosis is incredibly important.
Okay, thank you. And just showing the international reach, we'll keep the Australian accents going. Mandy.
Okay. Mandy Spurtle, QIMR Berghofer. Just a question about the e-health linking to organisations with e-health or countries. Are you being proactive in seeking out which countries or organisations have e-health and are not members?
So I'll answer that in 2 parts. First of all, the e-health group is really, it's just starting, and so what we're doing at the moment is mapping particularly activities, and it's most active, I would say, in the US, UK and Canada in terms of aiming to link genomic data to electronic health records. But then we've also had a lot of conversations with India, for example, because they're really way ahead in terms of getting electronic health records for many millions of people throughout India. I would see the sequence of this will be, first of all, looking, to identify what we can realistically do, and particularly in partnerships with the other organisations working in this area. We'll pick a couple of projects which are bite-sized chunks where we can actually start to demonstrate the linking of genomic data into electronic health records. And it really, across all areas of the Global Alliance, one of the I would say now short to medium term aims is to globally reach more widely in countries that aren't already involved in the Global Alliance, and particularly each of those countries will have different levels of advancement in terms of applying genomics in clinical practice, or, as you say, from the electronic health record point of view.
But one thing that your question makes me realize, and I've been thinking during your talk, is now that the electronic health record question is beginning to be worked, it really should be the case that the companies, this is an American comment, I don't know, and the equivalents in other countries, but companies like Epic and Cerner, which control most of the US electronic health record market, should be in this room.
Yes.
If they're not, they should be invited. If they refuse to be, that wouldn't be good. But hopefully they are and will be. And we should push, as is being suggested, different countries, different companies, get all those stakeholders to make sure that they're included.
I didn't like to mention specific companies, but certainly we have been engaging in terms of discussions. We want to make it quite agnostic, though, so that the types of linking of genomic data into electronic health records is not going to be reliant on a particular type of electronic health record.
In the same way that we have different information companies in the room.
So we have coming to the microphone in the middle John Madison, who is leading this initiative and can give us a definitive answer.
So that last point is very critical, and Cerner and Epic and the pharmacogenomics vendors have been invited to attend and are participating in similar efforts and will be participating in this effort. So they very much need to carry the water for the implementations. Yeah, absolutely. So they are invited.
And just reinforcing a point that John made on the first conference call of that was we would be doing a huge disservice if we had 6 different initiatives coming from the user side, all trying to make different appeals and different requests of industry. So consolidate so that we can have one collaborative. On the country level, certainly through things like G2MC, we're bringing in countries like Sweden, Estonia. We got some Middle East representation, so there is that country perspective. Mark.
And just one thing to add to Kathryn's excellent presentation. One of the things we've found working together, obviously I'm in the cancer space, Kathryn is in the rare diseases space, is how much we can learn from each other. And that's been an actual added value thing of the whole initiative, is actually learning from each other. Things that are, we use in one area can be very applicable in the other area, but maybe we just hadn't realized it. And that gets back to what David said earlier about the whole reason for this working is actually we learn from each other.
Okay, one more question.
This is Melissa Handel from OHSU in Portland. I lead the Monarch Initiative, and we've made great strides in making a lot of the human phenotype data interoperable with the model organism data for rare diseases and undiagnosed diseases. And just to this point earlier, I think that we really need a working group to further synergize the relationships between the cancer genomics and expansion of the human phenotype ontology and integration with other kinds of vocabularies and standards to help support that because we have so much crossplay between those two things. So maybe that's something that we can talk about. I know we have Warren Kibbe here, so maybe he could also speak to that, because we've been sort of going back and forth with NCI about that as well.
Okay, thank you. Warren, did you want to say anything on that?
Sure, I think it's really important that we do talk about how we get interoperability between model organisms, where we're really learning from what we've spent an enormous amount of time and effort in, and making it relevant, much more directly relevant to human disease and exploring it. And I think it's putting all these things together, and I think that's what we're all trying to do.
Okay.
I think
Thank
that's a
you.
fantastic point, and it came up in the metadata task team meeting of the data working group yesterday, is particularly not to be too humanistic, to actually look at model organisms and how we can integrate it, particularly from Keith's talk this morning of the various levels of evidence that we want to be able to link to a particular genomic or metabolomic or proteomic change.
Okay, I'll
Martin?
take one more question from Martin. Oh, no, we'll have one from Nicole and one from Martin. Go ahead, Martin.
So this is short, and I'm way off patch, Kathryn. One of the words that hasn't been raised at all so far is infectious disease and microbial genomes. So is that going to be within your area of interest, or are we ignoring it completely, or where are we going to pull that into the pot?
We put it on the back burner at the March meeting. It did come up as a discussion point there because of the Global Alliance of Infectious Disease. We thought we'd start with the human genome and then work outwards, but I would see that it would be incredibly important for interlinkages, but also when you're looking at, again, patient data and the interaction between environment, including infectious organisms and genomics.
So I think we,
David?
I think we need a group to look at this. I just would report that there was a meeting at NIH in September in which I was approached by some people from NIAID, the institute that does that. They're very interested in this. Had a meeting at one point with the Gates Foundation. They were interested in infectious disease more than some of the other things. I think that we made a conscious decision a year ago not to do it because we just had a lot of things to do. I think as we take notes of the things coming out of this meeting, I think the time is now. We should probably start by having a group that says, look at what we could do rather than go do it. So I think if people in this room, either themselves or know of people, I've had some discussions. Dom Kwiatkowski was interested in this, who'd be a great person. Maria, NIAID, others, but if people in the room can suggest names or volunteer, we could get someone to look at it and then hopefully within a month or two come up with a plan. I think the time is good.
And we have had one conversation with GMI as they're having their discussion. So. Nicole.
Thank you. Nicole Boyce with Global Genes. We're a global advocacy organization focused on rare disease. And I have a quick question. So, we know now that there's, a pretty significant shift in how some data, especially from rare disease, is being aggregated. And a lot of it's coming from the patient community that are early, helping fund early stage research. And they're now becoming more equipped and understand the importance of owning their data and then where they want to put it. So my question is, how do you plan on messaging and getting, the patient community, the global patient community involved in this, effort of collecting this very important data so that you all can use it?
No, excellent question, and part of that will be covered in the panel session this afternoon. Brad Margus is part of our steering committee, and we do have a big focus on public engagement, and I think that now that we have things that are up and running with which we can engage community organisations and patient advocacy groups, that really will be in the next wave of work that we're concentrating on, not just communicating how to use a particular API, but really what is available and what are the standards, particularly for collecting data and specimens that the patient organisations are also involved in. I would also note that there are a number of patient advocacy groups that are members of the Global Alliance as well, and consortia like IRD, IRC have very big community engagement.
Okay, thank you. I don't see any more questions, so Kathy, thank you.